Hi everyone, and welcome to our summary on cloning in plants. What we're going to be doing in this video then is having a little look at the spec reference 6.2.1b, the two parts of it there, looking at how we produce artificial clones in plants, and obviously these arguments for and against artificial cloning. In terms of how we can actually carry out this process of artificial cloning, then we're going to go through the processes that you need to know, because these are bits that can come up as practical based questions as well. So first option, we can take cuttings. Now, in order to take a cutting from a plant, it's quite a simple process. What we're going to do is we first of all select a healthy shoot. Obviously, you're not going to pick a diseased shoot on your plant, I would hope. So you select a healthy shoot, and then we're going to cut the stem at a slant between two nodes. Now, hope you remember, a node is basically the joints that we have for our leaves. So we cut that stem at a slant between those two nodes. We then dip our little cut stem into some rooting powder. And rooting powder is basically a little powder that contains some hormones for our plant, like auxins, which are going to help stimulate the growth of the roots there. We then put that little cutting into soil and we water it. Now, one other thing that we tend to do here to just promote the growth of this little cutting is we then cover that pot that we've put our little cutting in with a plastic bag. Or we could remove some of the leaves. Now, the end result of both of those things is the same. We're doing that to reduce the rate of transpiration. Now, transpiration, if we think back, is going to be the water loss, so the loss of the water vapour from the aerial parts of the plant, that definition that we hopefully know. Now, if we imagine that our new little cutting doesn't have a root system at this point, we don't want to risk our plant losing a lot of water, because it doesn't have the facilities to then take up large quantities of water. So if we can reduce transpiration, the water loss, then we're going to give it a better chance of survival. If they give you a question about how would you take a cutting, then these bullet points are usually corresponding to marking points themselves. So make sure that you can describe how to take a cutting in practical terms. We can also take cuttings from other sections of the plant as well, not just that stem. You can take leaf cuttings, where literally we take a leaf and we plant that in the moist soil. We can do root cuttings, where basically we take a little section of root and bury it just under the surface of the soil there. Or we could take scion cuttings. Now, scions are, as we can see this little picture on the right here, a little shoot or twig that contains buds from a woody plant. Okay, so depending on what plant we're actually trying to take a cutting from determines our technique. If they're talking practical based, then it's always going to be the stem that you're using there. So the practical method we just went through, that's the only one you really need to know. Just be aware that we can use other bits of a plant for cuttings too. The next option for how we can clone plants artificially is in what we call a tissue culture. Now, when we talk about tissue culture, this is where we're going to grow new tissues, organs or plants from some tissues which we've cut from the sample plant, the one that we want to basically clone. Two things we're going to need to achieve tissue culture successfully. First, we need a nutrient medium, and secondly, we need sterile conditions. Because we're not having a whole plant or section of plant that we're taking, we're literally talking about little bits of tissue that we're beginning with here, then that means they're not going to have those same systems for obviously getting things from soil. So we need to give them the nutrients from a medium that we are going to then grow those tissues in. The sterile conditions are going to be very important because again, they don't have the full range of defense mechanisms that a full plant has. Therefore, they are going to be susceptible to contamination from other organisms. Because we've got a nutrient medium, obviously that's providing great growth conditions for other things. We need to keep it sterile, so the only thing growing is the tissue we intend. Now, what we're going to do in order to actually get the end result that we desire here is that we will probably be adding things called growth substances at specific points in the development of this tissue culture. And the reason for adding these different substances is because it encourages differentiation. Now, differentiation is obviously where our cells are becoming specialized into certain types of cell to carry out a particular function. Therefore, what we're going to need to do to actually grow an entire plant is have differentiation take place. So using growth substances at the points through the tissue culture allows us to achieve that. When we talk about micropropagation, this is basically where we're going to grow large numbers of new plants from the meristem tissue that we've taken from our sample plant, the one we want to clone. Now, the great thing about meristem tissue, obviously we know it's a version of stem cells, therefore it can differentiate into a whole range of different cell types, but it's also free of viral infection. This is one of those unique bits, if you like, about meristem cells versus other cells in the plant. If you take cells from a leaf or the stem, there's a risk that they could have been infected by a virus. The meristem tissue will not have viral infection. Therefore, you are guaranteed a virus-free growth. What we're going to do now then is go through this process of micropropagation. So again, a technique that you need to be able to describe how we would carry it out, and you need to know some of these key terms that are going to crop up through this little description of the method. So first thing we're going to do is we cut our plant material from either the axial buds or the shoot tips, and we cut it into small pieces. Now, those small pieces of our plant material we refer to as explants. One of those keywords we need to know there. Once we've got our explants, we need to sterilize them. Now, the way we do this with our plant material is we're going to use a dilute bleach solution or ethanol. The whole purpose of that is just to kill any bacteria or fungi that may be present on the plant material itself. So we've got to sterilize the explants to avoid any of these potential contaminants from being present as we move into this next stage. Once they're sterilized, we can place those explants onto the growth medium, and this growth medium is going to be sterile itself first of all, and it's going to contain all the bits that our little explant needs to grow and develop. So we're going to have our glucose, the amino acids, the phosphates, all those kind of basics to provide us with anything we need to produce our biological molecules. And then we're also going to see things like the auxins and the cytokines. Once our explants are on that growth medium, they're obviously going to undergo the process of cell division through mitosis, link back to module 2 again there on the process, and they're going to form this structure called a callus. Again, a key word to make yourself aware of. Once we've got this callus, which is basically kind of a cluster of cells, what we're going to then do is we divide that callus into smaller clumps of these cells. Now, at this point, these cells are undifferentiated. So all we're doing at that point is breaking up this one lump of cells, large lump, into smaller lumps of cells. All of them are going to be genetically identical, remember, because they've divided by the process of mitosis. But that means we've now got numerous versions of our original plant. What we then do is we move those clumps to a new growth medium, and this is then going to contain materials which stimulates differentiation and continued growth. Now, depending on what we put them in, that's always going to differentiate into a different type of cell, or it's going to stimulate a different aspect of their growth. So what we do is we basically match the different conditions we're placing those little clumps of cells into to cause the roots to grow, to cause the actual stem to grow, etc. We keep doing this, obviously transferring them into their different growth media until they've developed into these teeny tiny little plantlets, and then we transfer them into compost. Once they're in compost, water it, grow it in a greenhouse, because that allows us to control all of those external conditions that could affect the growth of our plant to obviously maximize survival rates. So we've mentioned a few key bits there that we just need to be aware of, the kind of like the whys of why we did those steps. Now, obviously we talked about taking the tissue from things like the axial buds, because that's where we have meristem tissue present. Now, meristem tissue, as we know, is basically stem cells in our plants, and it is what we refer to as totipotent, which hopefully we remember means it can differentiate into all cell types. The explant, we sterilised it if you remember, and that was to kill the microbes, because if we didn't do that and we just placed it onto our agar, then it would just cause those contaminants to grow really well at the same time. That leads to competition. The contaminants would then be stealing the resources intended for our explant. It wouldn't end well. Obviously, we mentioned the fact that we've got all these different substances like amino acids, etc. in our growth medium, and that's to provide all of those nutrients necessary to provide the biological molecules for the growth of our plant. So amino acids needed to make proteins, etc. We mentioned the fact that there's also these auxins and cytokinins. They're all to do with stimulating growth of our plant. So auxin is one of those things you should be very familiar with from GCSE as one of those growth hormones in the plants. And as we mentioned, the greenhouse is then used to get those ideal conditions to actually allow the planters to acclimatise to growing under those normal conditions in soil before we just launch them out into the big wide world and they have to cope with the environmental changes that happen as the result of joyous British weather. So we now need to consider the advantages and disadvantages with regards to this artificial cloning in plants. First of all, let's have a little look at the advantages. So why would we want to carry out artificial cloning? Well, first of all, it's more rapid than growing plants from seed. So we're not having to wait for that process of germination, etc. So this allows us to get our plants faster than if we were using a packet of seeds. Secondly, we can actually reproduce plants that are tricky to reproduce sexually. So plants like orchids, for example, are a little bit of a pain when it comes to trying to get them to carry out sexual reproduction. We also have other plants that when we've got seeds, they don't always germinate. You've got to have the perfect conditions for them to actually want to grow from a seed. And this whole process of our artificial cloning kind of avoids all of those issues. So we can reproduce these plants where sexual reproduction is not viable or they're hard to grow from seed there. Thirdly, you can do it at any time of the year you like. Because we're obviously just taking cuttings or we're taking our little tissue cultures, etc., then we can control the environment and then just grow these plants whenever we want. We're not having to wait for seeds, we're not having to wait for them to germinate, we're not having to wait for the right conditions for all of that. Therefore, year-round growing is possible. We've already mentioned this in terms of cloning and the natural ones, that they're all genetically identical. Therefore, they're all going to share those desirable characteristics, which means that from a kind of a commercial standpoint, we can predict their shape, their size, their colour, whatever it may be, very predictable, which is beneficial to us. If we're going down the agriculture route, then that uniform phenotype is going to give us a much easier harvest, because if they've all got that identical genotype, phenotypes are going to be the same, therefore they're all going to display the same characteristics when it comes to harvesting those plants. And they're virus free. We're not obviously relying on seeds at all, and therefore we can grow our plants without the worry of viral contaminants. As always though, there are disadvantages to any process we have. Firstly, it's expensive to set up a tissue culture. So there is a greater cost involved there, and you can obviously see why from the process than just growing from seed. It's also a labour intensive process. If you're thinking about all those different bits that are going on throughout the process, then that's going to require more people doing more parts rather than just plant the seed, water it every so often. We do have a risk of failure if we do get any microbial contamination in that process of micropropagation, because any contaminants within our nutrient medium will obviously lead to competition, and that can obviously cause the whole culture to fail. And because they're all genetically identical, that means that they don't have variation. Therefore, they're all susceptible to the same environmental changes that could be harmful. The last bit I would suggest that you make yourself aware of on this little section of our topic is the kind of differences between our cuttings and our micropropagation techniques. So both of them are obviously producing artificial clones in our plants, but they could ask you to compare these two techniques. So just giving you three kind of key comparisons there. If we're thinking about cuttings, less equipment is needed than our micropropagation. Obviously, all we need to do is basically have something to cut our plant, a pot, some compost, a bag, and some rooting powder. Micropropagation, we're talking about different growth mediums, we're talking about different hormones necessary to cause it all, plus much greater control of conditions, petri dishes, you name it. Secondly, cuttings have less skills required in terms of the people carrying it out. To be honest, most of you could probably wander out in the garden and take a successful cutting, knowing not a lot about plants. Would you be able to carry out micropropagation so successfully? It's unlikely. So we need more skilled people involved in micropropagation than in taking cuttings. And the third one on there is that cuttings produce fewer clone offspring than the micropropagation. Because if we think back, once we've got that callus, we can split it into a range of different versions, and therefore we get more of our little genetic clones than we would through a cutting. As always, don't forget to subscribe to the channel so you can see when I upload another video for you. And of course, head on over to the website to get access to a range of other resources that should support you in your study of your A-level biology.